You have fallen into Event Horizon with John Michael Godier. In today's episode, John is joined by Dr. Łukasz Szydłowski. Dr. Łukasz Szydłowski is a researcher at the Sanno Center for Computational Personalized Medicine, specializing in bioinformatics, biotechnology, synthetic biology, and microbiology. He earned his PhD in chemical engineering and biotechnology from the University of Cambridge in 2015. His research focuses on microbial adaptations to extreme environments, including space conditions. His work has significant implications for understanding microbial behavior in space habitats and ensuring the safety of long-duration space missions. At Event Horizon, we know professionals like you demand high-performance, reliable solutions that fit seamlessly into your workflow. Unlike John, who's still trying to figure out how to be professional. But hey, we love him anyway.
Wait, what?
Meet Kinsta. Kinsta isn't just web hosting. It's a premium platform built for businesses that rely on their WordPress sites to drive operations and sales. Forget worrying about site maintenance, security, and speed. Kinsta handles it all so you can focus on what really matters, growing your business. And no, John, your assorted collection of white elephant knickknacks doesn't count as growing your business.
Huh?
Switching to Kinsta can boost your website speed performance by up to 200%. No extra optimization needed. Plus, with SOC 2 certification, you get enterprise-grade security, firewalls, DDoS protection, and automatic SSLs to keep your data and customers safe. So safe, not even John could mess it up.
What?
Managing your site. A breeze with MyKinsta, a custom-built intuitive dashboard that makes old-school control panels, like cPanel, feel ancient. Need help? Skip the chatbots. Get real human support from WordPress experts. Available 24/7, 365 days a year. Unlike John, who's only available after his third cup of coffee.
Oh, well, that sounds fair.
Right now, Kinsta are offering new customers their first month free on select plans, plus unlimited expert-led migrations at no cost. Go to kinsta.com/first-month-free to get your first month of hosting for free and to migrate your website over at no cost. And yes, I've never been able to get John's attempt at a website out of my head. He should have used Kinsta in the first instance.
Dr. Łukasz Szydłowski, welcome to the program.
Thank you for invitation. Welcome.
Now, Doctor, in closed environments like the International Space Station, where we have humans carrying our microbes up to low Earth orbit into an enclosed, sealed space, and those microbes are going to get out and they're going to do something. Now, what exactly do they do? Where exactly on the ISS do microbes colonize when humans go there?
Well, they kind of spread around the entire interior of the ISS, as the microbial tracking mission has revealed. So different parts of the station have been sampled. The swabs have been taken from surfaces, revealing the different amounts of biofilms growing on the, especially in the parts frequently visited by astronauts.
Now, is it places like the kitchen?
Yes, it's a kitchen, it's a crew cabins. Obviously, toilets would be one of the places where they do show up, as well as gyms and other places where people do leave traces on themselves, because, as you know, we cannot really sterilize astronauts as they go to the ISS. Therefore, all of them, they are actually each one astronaut is an entire ecosystem full of their own microbiomes that can also be dispersed and become a part of ISS.
So these are very human microbes. We're familiar with these here on Earth, but when they get to the ISS, something may start to change under the unusual conditions that it is present there. Now, are there these microfilms, are there different biomes in different parts of the station? In other words, is the kitchen different than the bathroom?
They do vary slightly. and that also depends on how how frequently those places are being visited. So those that are most crowded would share usually more or less the same microbiomes composition. However, what has been noticed was that the more harsher sterilization, cleaning procedures have been implemented to remove those biofilms, then what was observed was the growing numbers of bacteria that potentially be opportunistic pathogens. So those biofilms do have a tendency to reach some stasis balance in its way, and if we do interfere with it, we may actually get something opposite of what we would expect by removing them.
Now, this includes sort of gain, some kind of gain, that it starts to behave a little bit differently than it does here on Earth. What is doing that? In other words, is it resistance to just the environment? Is zero gravity doing this, or do we have any idea what's changing these microbes?
Yeah, so in a way, it would resemble places like hospital when we see a growing problem emergence of those opportunistic pathogens coming up because of the way all of the surfaces are being treated. However, in the case of ISS, there is also the key objective of the microbial tracking was also to check what has been unexpectedly shipped into the ISS. So we were also searching for those hitchhikers that would not be part of maybe typical human microbiome, which of course is quite tricky, because we only start to explore the diversity of human microbiomes, right? So we can't be really sure, but there are also microbes that can survive conditions in the clean rooms already in the spacecraft assembly facilities and be transported there, either in the form of spores, so not really alive and active, but then once the conditions become favorable, they can come back to resume their normal activities, metabolically speaking. So what we found was that there were quite different organisms that we managed to isolate from the biofilm from the complex microbiome community, and we did manage to isolate single species using by bringing samples back to Earth and growing them in the lab.
What different challenges does the environment of the ISS present to a microbe that's different from Earth? In other words, there's got to be things like carbon dioxide concentrations, and again, low gravity, zero gravity. How is that affecting the microbes?
So that's a good question, John, because of course there are a few key conditions that would determine how those microorganisms can thrive. So one thing is that the increased radiation that they experience, and we already knew back in the days when when the Deinococcus radiodurans was discovered that there are bacteria that can deal pretty well with the high doses of radiation, even extreme doses of radiation, I would say. Another thing is, of course, as you mentioned, microgravity, which is to some extent resembling hyposmotic stress. So living in such conditions makes cells generally larger than when they are grown when the same species are grown on Earth, because they experience this different concentration of nutrients around them than that could end up in cells growing to the point that they would burst. And this was quite interesting. What we did find, apart from this size differences, was that all of the species that were discovered and studied had this mechanosensitive ion channels, which could be referred to pressure valves, kind of removing the excess of the osmotic pressure and thus mitigating the risk of cell burst. Another feature of living in space is of course much less food availability, as you also mentioned, like either in the form of the carbon dioxide that could be a building block for organic materials and generally organic matter in available. So those biofilms tend to grow slower than they would on Earth, given this oligotrophic setup, but they do show some more diverse metabolic pathways. in a way that they need to cope with this by the food shortage by trying to digest whatever is available to them.
Now, does this present a challenge medical wise? These mutating and evolving bacteria on the station, does this present a problem for microbial problem, not really a viral problem, I would imagine, but a microbial problem for astronauts on the ISS?
That was exactly the reason we did conduct those analyses. We were very much interested in looking into how those adaptations could pose a threat to the human crew of ISS. And we, of course, we didn't have in reference to those species studied, we did not have the time zero, and we did not have the population of terrestrial organisms before they entered ISS and were launched into space. So we kind of assumed that they might be there as long as ISS is operating. And luckily for us, or for astronauts up there, none of them had any indications of being potentially dangerous or resistant to novel antibiotics. Actually, we found quite interesting feature in one species named Arthrobacter butitsky. It was found that it contains a pathway encoding novel antibiotic family. So the fact that they have to survive in a very harsh environment, which puts a lot of selective pressure on those microbes, means that we could actually use that potentially to fight them off if any of them would turn out to be pathogens. So that's very that was overall quite exciting finding to see that we could probably be able to find new ways for therapies even on Earth for antibiotic resistant bugs.
So let me make sure that I'm getting that straight. So the fact that these microorganisms are being forced into increased novelty, I guess you might say, that is also making them, with a specific family of antibiotics, actually more susceptible to being able to treat it. Did I get that right?
Yes, that's right. And we also found that in the population of bacteria that were sampled from the spacecraft assembly facilities, there were also novel families of genes encoding those short peptides called lasopeptides, which are also antibacterial in their properties, and they are very specific. So they can target very specific species or even strains of bacteria. So we could selectively remove some unwanted members of those microbiomes should we need that, and that's very good, because what we see is that with limiting the diversity of microorganisms, we also see emergence of those unwanted, potentially dangerous species. So it would be great to have those alternative methods to mitigate risk of outgrowth of pathogens, whereas retaining the overall population of the naturally evolving microbiome that is neutral, or even could be potentially beneficial for us, or for astronauts, especially in the context of long-term missions.
Now, we have done experiments on the ISS and other places growing plants in space, and that's ultimately the goal there is food. But the thing is, you need a microbiome in the soil usually for that. And some species, for example, nitrogen fixers like beans, carry them with them. So could we use this information that we're learning from your studies to tweak, these biomes better for the plants than they would normally be in space?
Of course, actually, that's a part of my interest that has been taken beyond the scope of this research and context of the startup that me and my colleagues founded, which basically focuses on developing these biological solutions to enhance plant growth on exoplanets like Mars or Moon. So this is actually a very good starting point to start thinking about it, because if we want to colonize other planets, definitely we need to know what we are bringing there, and we need to plan it carefully so that we could enhance this process of releasing the nutrients that are available in regolith. However, they're not so easy to be absorbed by plants without the help of these symbionts.
The idea, though, of these enclosed spaces becoming sort of incubators of sorts, abnormal incubators for microorganisms, we're going to be sending a lot of those sealed environments out, especially if we start putting human boots on Mars, in whatever form that will take. And it takes a while to get to Mars, and that's a while for bacteria to mutate and do their thing. And my question here is, are they always going to do the same thing, or is each enclosed environment going to end up different?
Well, that's actually a very good question, because of course we are dealing with completely new conditions that they would encounter outside of our planet, and those conditions of course do stimulate the higher mutation rates and adaptation towards new environments. So what we would design them initially to do, that might be tricky to maintain in the long-term scale. So especially if we are dealing with decreased diversity compared to what we have on Earth, right? Because we can always dig up for some more microorganisms. We can enrich some members of these complex communities, whereas on Mars we'd have to start with what we have, and probably there would be, of course, there would be conditions promoting the development of new species, but we may also lose some of them in the process, and whether intentionally or not. And this is something that we don't know how the dynamics of it, how it would happen over the time.
Seems almost dangerous in a way.
Yeah, exactly. That's also something to consider all the time, that we may unintentionally create some superbugs that would turn out to be threats. So it's always good to know how we could remove them if we have such a need.
Now, this seems like it's somewhat related to a little bit further out there idea, although not, the idea of panspermia. In other words, microbes hitching a ride on a meteorite, an asteroid or whatever, and being deposited elsewhere, whether Mars or Earth or wherever. Could this process be happening inside those meteorites that might be transferring spores?
Well, when they are in the quiescent form of spores, when no activity is taking place, it's really hard to say if any changes occurred there, because it's only after they're trying to restore themselves to life forms is when you can have this rearrangement of the genomes, or horizontal gene transfer at the higher rates that could promote the diversity. But it's quite interesting to see, like, given that, for example, how easily it was to contaminate Ryugu asteroid with bugs, how prevalent life is, and how much it can thrive even under those unfavorable conditions. So I think that the panspermia theory, it's quite very likely to be true, and to be how life could spread itself around solar system or even beyond.
Nuts and bolts of retrieving bacteria off of the International Space Station. What did the methods look like? Did you have astronauts swabbing, areas, taking samples, or were you bringing back pieces of equipment and a return capsule?
So,
what were you doing?
During microbial tracking mission, they were basically doing swabs and packaging those swabs, in sealing them, and then sending them back to Earth. So not much was done on board of ISS, apart from just collecting the samples and making sure that they are safely preserved until being shipped back. However, right now, as I know, there are many more tools being available for astronauts to carry out more complex molecular biology works, sequencing them on board, and that's quite exciting to know that they can discover new species on the go as they encounter them from those biofilms that are present in ISS, because it took some time to ship the samples back. So of course you could also argue whether some of them were lost, and of course not all of the species can be cultured on Petri dish in the lab. So that is something that will hopefully be done on the larger scale in the coming years.
DNA repair. Now, one of the hallmarks of getting microbes and radiation together is their ability to repair their DNA. Mm-hmm. Did you see an enhanced ability in these species to repair the damage?
Oh yes, yes. All of them had very diverse systems for both homologous repair as well as non-homologous end joining. So that was a common feature for all the 5 studied species that we did describe in the paper, which was quite interesting because they were all different species. There was one spore forming for non-spore formers, which was also kind of counterintuitive because I would expect more spore formers to be present in such conditions. But yes, this DNA repair systems are very robust, and they allow them to fix both single-strand DNA breaks as well as double-strand DNA breaks. With this, with the latter being more damaging because, but then at the same time, if upon repair, it also it allows higher rate of horizontal gene transfer because the templates used to seal the discontinued double-strand breaks can be coming from other organisms that are around.
Now, it seemed to me that now this also gets into the appearance of genes, and it seemed to be quite a lot. I found that part surprising. Was that you start seeing genes appearing? What is the mechanism for that?
So the genes that are present there that have not been found in their closest relatives are quite interesting because they were the ones that we thought would be the source of their adaptation to the ISS conditions. But it's quite diverse given that we don't know much about their closest terrestrial relatives, and we did not have a ground control for when those particular organisms took the ride to ISS. So we don't know how much of that gene divergence took place and when did they appear, whether they were already there and that's what allowed them to be the astral hitchhikers, or whether they did show up in the process of adaptation to the extraterrestrial life.
Is it possible, and this is an off-the-wall question that I may be extremely off base on, but you'll explain it to me, could you have an inactive area of the genome of a microbe suddenly become active because it's suited for this environment? In other words, could what we would normally call junk DNA, although I'm very careful with that term these days, could that something reactivate that was there from long ago?
Well, especially in bacteria, the prokaryotic genomes tend to be smaller and much free of this kind of non-protein coding part of DNA. So it's hard to say whether there could be all of a sudden an instance of inactive part becoming active. It's more of a repurposing of the genes or gaining function by through a series of mutations. We also saw, but not in all of those species, definitely saw some prevalence of potential phages genomes. So there could be some, let's say, foreign parts of the genomes that could all of a sudden be used, and also mobile genomic elements like plasmids and transposons were also there, but not in equal way. It wasn't like equal feature shared between all of them.
Could it be indicative, and again, this is maybe even more off base, that maybe some of these adaptations might have been there, or remnants of them, or whatever that could be used, because that microbe had been in space at some point before, knocked out on a rock and fell back?
Yes, it's always a possibility. However, what we don't know is that obviously we were dealing with the isolated genomes of 5 of the species, but definitely what we were missing in this image here was the context of the microbiome that they were thriving in. So we don't know exactly what was their neighbors in their biofilm, what was their interactions between other species, how did they interact. So it was more of a kind of opportunistic pick of the species that we were able to grow in the lab. And of course that brings another question: how do they form this ecosystem? What are their typical interactions or lifespans if they respond to one another, or are they competitors or symbionts? So this is something that is definitely worth exploring more in future work.
Now, a question about trends with the ISS microbes is the idea of, on Earth we have convergent evolution, where you end up with, and it's in macroorganisms, there's a reason a dolphin looks like a shark. And is this occurring on the ISS? In other words, is there a trend for all of the species that you found
Mm.
moving towards the same sorts of adaptations?
From the point of view of those studied species, we see some in some cases the convergence. Like for example, this mechanosensitive ion channels, they do definitely appear in all of the studied species. So that's a hallmark of definitely something being needed regardless of what species you are and how many genes you have. Other things would be, I would say definitely there's a whole DNA repair kit. So that all of the genes involved in those systems, they are pretty much consistent in numbers. So it seems that there is definitely this common feature shared between them. But in terms of convergence in other traits, it's hard really to say, because some of them did produce carotenoids, which we think that might be important in terms of radiation protection, but not all of them. And as well as those kind of secondary metabolites like the novel antibiotics, it's not a shared feature between the species.
Now, how does CRISPR relate to your work? What can you do with that?
Well, CRISPR would be really nice tool to actually edit the genomes on the large scale, trying to pinpoint those critical adaptations of what we think are critical genes, and either knock them out or transfer them to organisms that we know would be susceptible to such conditions, and see if we can obtain another species and precondition them to live in ISS, or we can switch off this ability from the species that are there. That would be definitely good application of CRISPR technology.
Now, does this present a possibility of learning something about new medications here on Earth? In other words, by studying these bacteria and how they're changing, would this also perhaps benefit us down here that might lead to new drugs?
Oh yes, of course. I'll definitely be eager to look into those pathways in more details, because we only been able to describe them bioinformatically, but more tests in the labs are needed, and we could definitely see if those could benefit us on Earth. I'm pretty sure they could. So these findings can be definitely beneficial, not only for astronauts, but also for terrestrial populations.
Now, a further question about living in space is the idea of artificial microbes, or very, very attenuated microbes or altered microbes, to help deal with things like getting rid of trash and getting rid of carbon dioxide and things like that. What's the landscape of that area of your field look like right now, as far as figuring out what can we do with custom microbes?
Well, that's a very good topic to explore, because the whole area of synthetic biology is super happy to kind of work with such microbes by design, right? Given that we do need to carry out terraforming if we ever want to be permanently living in places like Mars. So in this case, having adapting specific organisms would be crucial for carrying out certain transformations or certain activities, like releasing minerals from regolith. And this is something that is already people are looking into ways to enhance that, because of course we have some microorganisms that can naturally do that. We do see their abilities from the terrestrial populations, but being able to increase that is very, very important. Another thing is that we are also trying to think about it in the context of next level of complexity. So not only about single organisms, but how to put them together in a kind of working network, right? So we would need to design carefully a lot of different components of that network, so they could benefit each other, and by that also retain those features for a long time as we need them.
So in other words, it would be an entire artificial biome.
Yeah.
Basically
Yeah. So I think that pushes this synthetic biology, which is already operating in kind of synthetic communities or designed communities, into kind of designed biomes, and that's something that requires a lot of modeling, a lot of understanding of what we need for that system to be sustainable.
Now, my last question for you is this: what is the future of your research with the International Space Station? What's next as far as studying these organisms, and what's the roadmap for deepening this research?
So I definitely want to focus on two things. Learning from the microorganisms that we know that can adapt to those conditions, how we can use them for for future extraterrestrial applications, like promoting plant growth and food production in places like Mars, and also what we can do to prevent what I call a Martian scurvy. So any possibility of the outbreak of any kind of disease caused either by malnutrition or accumulation of unwanted things in our organisms that could lead astronauts to become sick and incapable of surviving on another planet. So making sure that this would never happen by designing microbiome-based solutions for astronauts' well-being and for future colonizers, like kind of survival packs, is definitely my goal for the future.
And I think it needs to be done now, because it certainly looks like we're going,
Definitely.
either to Mars or the moon. So
Exactly.
we need to know now.
Exactly, and that's I'm so happy to live in such wonderful, interesting times when opportunities like that emerge.
Absolutely. Thank you, Doctor. And I hope as your research progresses, you'll come back and visit us and update us on what else you find about microbes in space.
Thank you, John, and with pleasure. I'll definitely be happy to share my findings in the future.
Event Horizon and my channel are now available as a podcast on Apple Podcasts, Spotify, and YouTube memberships. Early ad-free episodes, bonus episodes, and sleep-focused content. Sign up now by clicking the links below to your platform of choice.